The overall goal of this procedure is to visualize structures such as F-actin in the NK cell lytic synapse using STED microscopy. This is accomplished by first recapitulating the synapse on glass using activating antibody. The second step of the procedure is to fix, permeabilize, and stain the cells for structures of interest. Next, the slides are imaged by laser scanning confocal microscopy, followed by application of STED depletion for increased image resolution. The final step is to process the images using a deconvolution algorithm. Ultimately, results can show sub-100 nanometer resolution of cellular structures thanks to dual-color STED nanoscopy.
This method can help answer key questions in the cell biology field, such as the relationship between the cytoskeleton and exocytosis. In our case, we are interested in the secretion of specialized lysosomes termed lytic granules by natural killer cells. Generally, individuals new to this method will struggle because there are multiple variables that can be adjusted in the application of the STED depletion laser. An inappropriate application of the depletion beam will result in a worsening of, instead of improvement in, resolution.
In preparation, warm 30 milliliters of RPMI media with 10% FCS, and separately 1 milliliter of BD Cytofix Cytoperm, both to 37 degrees Celsius. Then begin the process of coating the cover slips with antibodies. Take number 1.5 cover slips and, using a PAP pen, make dime-sized squares on the cover slips. Prepare one square for each tested condition.